Jessica, and I'm so excited today because we will be discussing translation. In the earlier process called transcription, we go from DNA to RNA. Translation is the final step that leads into protein synthesis. In this lesson, we're going to learn where translation takes place. We're going to understand how mRNA, along with its friends, the tRNA and ribosomes, work together to produce proteins, and of course discuss the stages of translation in protein synthesis. So let's get started, everyone. So what is translation? Basically, it is translating from one language to another. In this instant, a messenger RNA is translated or decoded, and its information is used to build a polypeptide or chain of amino acids. The key components required for translation are mRNA, ribosomes, and transfer RNAs. The messenger RNA, mRNA, is a single-stranded molecule that complements a specific DNA template. And as you can see, right next to it, we have its DNA template or complement DNA template. Now the difference between the two, as you can see, it's the DNA is double-stranded and the RNA is single-stranded. So the mRNA is the final product of transcription and is made up of codons. Each codon contains a chain of 3 nucleotide bases specific to a particular amino acid that will be added to the polypeptide sequence. The mRNA interacts with the ribosome RNA, recognizing the start and the stop codons present within it. So as we go along the ship, we will come across the transfer RNA. The tRNA is made up of 80 ribonucleotides. It contains small sequences of 3 nucleotides called anticodons. These anticodons complement the codons of the mRNA. When the anticodons of the tRNA combine with the codons of the mRNA, the tRNA carries the amino acid and adds it to the polypeptide chain. So as we continue along the spaceship, we're going to come across the amino acyl tRNA. And this is basically an enzyme that helps attach amino acids to the corresponding tRNA. So as we continue on through this ship, we're going to come across ribosomes. Ribosomes are where mRNA goes after it leaves the nucleus. Now, this 3D model demonstrates that ribosomes have separate subunits composed of proteins and rRNA. Ribosomes read the nucleotide sequence from the 5' to the 3' direction when it binds to the mRNA, and it synthesizes the corresponding protein from amino acids in the N-terminal to C-terminal direction. So as we go on and peek outside the ship here, we will see the larger subunit of the ribosome. It contains 3 active sites, as you can see, the E site, the P site, and the A site. Now the A site is where the incoming amino acyl tRNA binds during elongation. The P site is where the tRNA linked to the growing polypeptide chain is bound. And the E site, also known as the exit site, is the binding site for the tRNA prior to its release from the ribosome and after its role in translation is pretty much done. So as we continue to walk around the ship, we're gonna come across this diagram or picture. This shows where translation takes place, and it actually takes place, as you can see, and we come a little bit closer, in 3 stages. You have initiation, elongation, and termination. So as we go ahead and we peek outside the ship again, we come across the first stage called initiation. So initiation basically is when the smaller subunit binds to the 5' side at the start of the mRNA. It begins to read the mRNA in the 5' to 3' direction until it encounters a start codon. Now remember, all mRNAs have the same start codon called AUG. It codes for methionine. The large subunit also attaches to the initiator tRNA, which carries the methionine binding to the ribosome's P site. Proteins called initiation factors are required to initiate protein synthesis. These three main initiation factors are IF1, IF2, and IF3, which are important, very important in prokaryotes. Now, below the initiation, you have the elongation site. Now, this stage occurs in 3 stages. The amino acyl tRNA binding, peptide bond formation, and translocation. Now, during the AA-tRNA binding, a second tRNA carrying the following amino acid comes into the A site and recognizes the codon on the mRNA. This binding is through EFTU, and it utilizes GTP for energy. GTP is then hydrolyzed or finished, and the EFTU GDP gets released. Now, this EFTU GTP enters back into the EFTS cycle. Then we have peptide bond formation. The amino acid present in the tRNA at the P site is transferred to the tRNA at the A site, forming a polypeptide bond. This reaction is catalyzed by the peptidyl transferase. The tRNA at the P site becomes uncharged. Finally, we come to ribosome translocation. After the peptide bond formation, ribosome translocation occurs in which the ribosome moves one codon ahead to the 5', 3' in the 5', 3' direction on mRNA so that the dipeptide tRNA appears on the P site and the next codon appears on the A site. The uncharged tRNA then exits from the ribosome and enters the cystosol. The EFG GTP, the translocase enzyme, is required in ribosome translocation. This changes the actual 3D structure of ribosomes and catalyzes the 5' to 3' movement. The codon on the A site now becomes acknowledged by the other amino acyl tRNA. The dipeptide on the P site gets transferred to the A site to form a tripeptide, so it's growing. This process continues and results in a long polypeptide chain of amino acid. So to sum up elongation, a charged tRNA enters the complex with every step. The polypeptide becomes one amino acid longer, and the uncharged tRNA departs while the ribosome catalyzes every process in 3 sites by shifting one codon at a time. So after all this is done, we're going to go and we're going to start heading to the final stage of translation. And here we go. Finally, if we can turn around here. Here we go. So finally, we reach termination, which is when basically we reach one of the 3 stop codons, UAG, UAA, and UGA, becomes positioned in the A site. The stop codon are recognized by the next protein called release factors, which are RF1, RF2, and RF3, which hydrolyzes and causes the release of all components. So the 30S, the 50S, the mRNA, and the polypeptides all separate. The RF1 recognizes UAA and UAG, while the RF2 recognizes UAA and UGA, and the RF3 dissociates the 30S and the 50S subunits of the ribosome. After several ribosomes complete translation, the mRNA is finally degraded to reuse the nucleotides in another transcription reaction. So it's like a circle, keeps on going. And I hope you guys enjoyed this video, and thank you once again for having me as your instructor.